This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 174, recorded on April 12th, 2018. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm well, and you? Everything well?
Hanging in there.
Hanging in there, that's what they say.
Which is easy to do in San Diego at this time of the year.
You've seen any mushrooms lately?
Actually, would you believe, we live in a retirement residence, and on the front, right by the street, is a bit of grass and mulch.
Mm-hmm.
And I saw morel mushrooms there.
Wow.
Which is not bad.
Is this the time of year they would come up?
Yeah, around here, while they come up. See, that's a place that's getting watered.
I see.
Otherwise, in San Diego, we got problems with mushrooms.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Got any mushrooms there, Michael?
Oh, we got loads. It's been raining, and they sprout like weeds. They come up, but we have the scary mushrooms that take out your liver. And so,
really, the Continarios?
Yes, I remember when I was first here, we had
Amanita. Wait a minute, no, you probably mean Amanita, don't you?
Yeah, Amanita that take out the liver. We had
That's
a
right.
tragedy where there were a bunch of teenagers who were out in the farm fields near Orangeburg, South Carolina, and they were picking mushrooms off of cow patties, and
I heard about that.
And the young girl, unfortunately, took out her liver. But fortunately, her mother was able to donate a piece of one of her lobes of her liver.
Mm.
And so both the young lady and the mother survives. But
wow,
it's very scary.
It was very scary. And the child was lucky the mother was compatible.
That's all of an account of alpha-amanitin, an inhibitor of RNA polymerase.
Yes.
That's right. That's how it works.
All right. Today, after our two papers, we're going to give away the copy of Clinical Laboratory Management. We have your emails, and we're going to pick one at random. And I also want to tell you, have a note from the organizers of the 19th Annual Microbiology Student Symposium at the University of California, Berkeley. They want you to know about their one-day event, which will be held on campus on April 28th. This is organized entirely by hardworking and dedicated graduate students and showcases student research through oral and poster presentations. Each year, the students vote on two keynote speakers who are leaders in the field of microbial ecology and molecular biology. This year, they are hosting Doctors Kelly Wrighton from Ohio State University and Melanie Blokesh from the École Polytechnique Fédérale de Lausanne in Switzerland. The organizers are always growing the symposium. They want to encourage any interested listeners who live in the Bay Area to attend. Last year, they had attendees representing 19 institutions from the Bay Area. Symposium is completely free, and they have a limited number of travel grants for students who need childcare or funding for their trip to Berkeley. Registration and more information can be found at nature.berkeley.edu/calmicro.
You know, the graduate students in Boston do something very similar, and I think it's soon. Unfortunately, I don't have contact information, but if somebody were to Google microbiology Harvard, they would find it.
Okay. They organize a similar meeting.
Yeah.
Those are generally the best meetings to go to because they're free. You hear the next generation talking about the science that they're pursuing, and often if they have a sufficient budget and they reach out to the quote named speakers early enough, they can get some really marquee type speakers to come in, and they sort of all let their hair down, and you hear some of the best talks you will ever hear.
It's true.
Well, check it out. All right, for you today, we have snippet and paper, and Michael Schmidt is going to give us a snippet.
Well, so today on TWiM, we have enteric fever and the gathering storm of whether or not an untreatable typhoid fever is on its way to a country near you. So in this snippet, we're going to talk about two manuscripts in a snippet. The first is by Levin and Simon from the University of Maryland's School of Medicine, and it's a commentary that appeared in MBio. And the second is the paper with all the data that also appeared in the February issue of MBio. So both of these are open source. And when you sit down to read these papers, I would encourage you first, if you're unfamiliar with the terror caused by enteric fever, to first read the commentary, as it provides a very sobering background that will help put the significance of the content associated with the primary paper into really effectively a shot heard round the world. And that's why I picked these two to talk about today, because it's really pretty frightening what's happening. So the commentary is by Myron Levine and Raphael Simon. And it's, as I said, they're from the Center for Vaccine Development at the University of Maryland. And it specifically comments on the data associated with the second paper, the emergence of an extensively drug-resistant Salmonella enterica cerevar typhi clone harboring a promiscuous plasmid encoding resistance to fluoroquinolones and third-generation cephalosporins. And this, again, was published in MBio on the 20th of February. And it's authored by Elizabeth Clem, Sadia
You don't want to
Sapor.
read them all. It's too many.
All right, I won't read them all. We'll just say Clem. But it's a really internationally recruited group of individuals from the Wanger Trust in the United Kingdom, the Aga Khan University in Karachi, Pakistan. And it's literally the who's who of these nations' infectious disease investigators. So here's the story. Clem and colleagues conducted a comprehensive antibiotic sensitivity survey in concert with genomic analysis of this particular strain of Salmonella that causes typhoid fever. And they did this principally from blood culture isolates during an epidemic of typhoid fever that was occurring in Pakistan at the time. As you might expect from the title, the microbiologic and genomic data suggests that this cerevar Salmonella responsible has become extensively drug resistant, or is now considered an XDR microbe. And XDR is just below the step where you become TDR, or totally drug resistant. And so the data with their paper supports
X stands for
extensively,
extensive drug resistant. Right.
Extensively drug resistant. And the same abbreviation is used for things like tuberculosis. And so the data within their paper supports this frightening scenario in which we might see the return to the bad old days where Salmonella typhi, and I'm just going to truncate it and call it Salmonella typhi rather than the god-awful taxonomic proper nouns with Enterica cerevar typhi. The bad old days, according to the CDC, typhoid fever is a life-threatening illness caused by this bacterium. About 5,700 folks in the U.S., even today, will come down with this disease each year, with the majority of the cases being acquired as a consequence of international travel. About 75% of the 5,700 are a consequence of this international travel.
The others are locally acquired, is that right?
Locally acquired, probably case contacts within the house, because, again, this disease is fecal oral. And if you're one of the unfortunate ones that becomes chronically infected with this microbe, you can continue to shed this organism fecally. And if you are preparing food for your family or working in a restaurant and you fail to follow proper hand hygiene, you can actually easily transfer this organism between people.
Typhoid
It's
Mary.
the story of Typhoid Mary, right?
And we're going to talk about Typhoid Mary, or we can talk about Typhoid Mary now, whichever you prefer. But I'm going to put Typhoid Mary in the context when we get through some of this background. Now, the WHO estimates that there are about between 130,000 and 160,000 individuals will actually die each year from typhoid fever. So this is up there with the number of people who die from hospital or healthcare associated infections. Now, symptoms include prolonged fever, fatigue, headache, nausea, abdominal pain, and I'm talking significant abdominal pain. And here's the wild card: constipation or diarrhea. It's either one or the other. And about 3 in 10 patients will develop the hallmark rash with these rose spots. And it's one of the classic presentations of rash associated with bacteria. And so I went out and picked up a picture for the show notes so you can see what these hallmark rash with rose spots actually look like. And the severe cases may lead to serious complications. Now, in the bad old days, before antimicrobials, typhoid fever had a case fatality rate of 15%. That's effectively only half of the death rate associated with smallpox, or in the modern era, something like Ebola, where the death rate is 30%. So Levine and Simon, in their commentary, set the stage of why we need to be worrying about this extensively drug-resistant variant of this microbe that has arisen in Sida, Pakistan. Now, the danger is that most clinicians and clinical microbiologists have forgotten this lethality that typhoid fever exhibited in the pre-antibiotic era, 70 years ago, effectively. On the surface, it comes across as an acute generalized infection of the gut-associated lymphoid tissue of our reticuloendothelial system, and it can actually infect our gallbladder. When I teach the enteric diseases to the dental students and the medical students, I always put diarrhea into 3 buckets. And the first bucket has the first bucket has the diarrheas in which you can literally ride them out on the toilet. They're typically self-limiting. It's an enterotoxic. You get an E. coli that is manifesting a toxin that isn't very significant, and you effectively ride it out, and you treat yourself.
Staff does that too, right? Staff
can do that as well. The second one is where you develop a true infection, and that you can note by cramping, and the diarrhea is a little bit more severe, and you may need antibiotics in order to get rid of it. And the third, this is where Salmonella typhi falls in, is the form of diarrhea that goes systemic. This is where the organism can invade our system, and this is effectively what Salmonella typhi does, is it invades through our reticuloendothelial system and can set up housekeeping on our gallbladder and do all sorts of bad things. You actually get a bacteremia, and in some cases, you can become septic. It was so bad that the Journal of the American Medical Association, or JAMA, would publish yearly updates and 5-year summaries of typhoid disease for the most populous cities in the United States. And in fact, this is the first reference in the Levine paper. And it's an anonymous report, probably from the editorial staff of JAMA in 1914, describing typhoid in the largest cities of the United States. Now, this all miraculously changes in 1948. And this is an interesting story that they, again, bring to our attention about Theodore Woodward from the University of Maryland School of Medicine, who was conducting a clinical trial to determine the efficacy of the antibiotic chloramphenicol to treat scrub typhus. Now, typhus and typhoid fever are different. Typhus is caused by an obligate intracellular parasite that's effectively a tick-borne disease. And scrub typhus is caused by Orienta tutsigama, which is a gram-negative alpha bacterium from the family Rickettsia. And what happened is that the people enrolling individuals into the clinical trial for scrub typhus inadvertently enrolled people who had typhoid fever. Today, those people, they wouldn't even have talked about those data. They would have been excluded from the data analysis of the clinical trial. But here's where Woodward said, never look a gift horse in the mouth. And what he learned from looking at these patients that were actually unknowingly enrolled is they responded well to chloramphenicol. And well, the rest, as they say, is history. Case fatality rates due to typhoid fever crashed from 15% to less than 1%. And it was like other diseases successfully treated during our golden era of antimicrobial therapy. It was wondrous. It was a miracle and highlighted all good things that were occurring subsequent to the end of the Second World War. What we witnessed over the next 70 years is, of course, how evolution and single drug therapy can lead to the emergence of resistance to the remarkable wonder drugs that we all came up with in the golden era of antibiotic development. So here's where we transition to in the Levine and Simon paper, where they describe the resistance patterns and the plasmids. And this is where it gets to inside baseball about resistance plasmids, incompatibility group plasmids. Now, while R factors could be indeed transferred into Salmonella typhi, they could never be stably maintained. And you never use the word never when it comes to microbes, because eventually they figured it out. And that's part of our story here. But for the most part, this incompatibility group H1 in the early 1970s finally acquired a resistance trait for chloramphenicol. And it was maintained on the Inc. H1 plasmid, which then resulted in a large epidemic in Mexico and Vietnam, and then a decade later in Chile and Peru. As this was the golden age of antibiotic, while resistance to chloramphenicol was devastating to the people afflicted, we soon had other antibiotics that were found and recommended to treat typhoid fever. And these, of course, were ones that you've all likely heard about, ampicillin, amoxicillin, and trimethoprim sulfa. Things again changed in the late 80s and early 90s of the last century with the emergence in Southeast Asia that Salmonella typhi is now harboring the heretofore unable to harbor R plasmid. But now this R plasmid is encoding resistance to most of the first-line antimicrobials. But this was still the era in which antibiotics were being brought to market. And fortunately, at that time, it was the introduction of the fluoroquinolone class of antibiotics, which inhibit another target other than the ones that were being targeted by these early antibiotics. And the target that ciprofloxacin and the other fluoroquinolones is effectively attacking the DNA gyrase. And the gyrase class of inhibitors were found to be this remarkable drug in that they were able to affect high clinical and bacteriological cure rates. And very few of the patients treated with the fluoroquinolones ever became chronic gallbladder carriers, whereas the other drugs, some of those individuals did indeed succumb and become chronic gallbladder carriers. And so either you had to increase the course of antimicrobials, or the therapy at the time was to take someone's gallbladder if you couldn't get it out with the antimicrobials. So this was really a wonder drug. And so indeed, a 4-week course of a fluoroquinolone was approximately 90% effective in curing chronic typhoid gallbladder carriers without the cholecystectomy or taking out of their gallbladder. Now, like with all antimicrobials, inappropriate treatment course and their widespread use led to selection of mutations in the chromosomal gyrase loci. So as we approach our millennium, we're faced with multi-drug resistant variants of the microbe. So here again, an antibiotic against a cell wall target, the cephalosporins, came into use, especially ceftriaxone. So as we approach current time, there were occasional reports of patients being infected with ceftriaxone resistant Salmonella typhi. But fortunately, none of these resistant strains ever started a large outbreak, nor were they endemic in areas where they were found. That's until now. And here's where Clem and colleagues fire this shot that's heard round the world. First, they describe a sizable epidemic of typhoid fever in Pakistan that's caused by this strain carrying an array of both plasmid-borne and chromosomally encoded resistance genes. Not one, but many resistance genes. And they can collectively transfer resistance to the majority of our first-line oral antimicrobials used to treat typhoid, as well as the IV-delivered ceftriaxone. The only remaining reliable oral first-line antibiotic left in our kit is azithromycin. Azithromycin, that many of us in the United States know as the Wonder Z-Pak, is this remarkable antibiotic that's acid stable, so you can take it orally without any need to protect it from gastric acids. And in fact, it works better on an empty stomach. And the remarkable thing about azithromycin is it's very active and accumulates or concentrates into phagocytes. And if you know anything about the biology of Salmonella typhi, it's a facultative intracellular parasite living inside our cells. And it can actually live in phagocytes. So consequently, azithromycin is this wonder drug because it can, effectively treat the cell in which the organism happens to be hiding. But in spite
I'm allergic to it as all get out.
Which
So I hope I don't have to use it. I couldn't use it.
Well, in spite of Z-Paks being our hope in Pakistan, what is most concerning is that we've seen resistance to azithromycin treatment have already been reported in Southeast Asia. So the nuts and bolts of their data is they sequenced and evaluated a large number of the blood culture isolates from the epidemic over this period of time that the epidemic was raging. And what they learned is this Sida strain carries a composite transposon that's integrated into the chromosome and encodes all these multiple resistances to chloramphenicol, because you can have more than one gene conferring resistance. So it's multiple resistance to chloramphenicol, amoxicillin, trimethoprim sulfa, and it carries a single mutation in the chromosomal gyrase A gene, as found in many other H58 strains circulating in Southeast Asia, that confers intermediate resistance to ciprofloxacin or Cipro. In addition, this particular H58 clone carries this INC-Y plasmid. Now, the INC-Y plasmid is a big plasmid. It's not like your garden variety cloning plasmid. This is 85 kilobases. And this plasmid's got everything in it except instructions on how to build a plane. I mean, it included plasmid quinolone resistance genes, the beta-lactamase CTX-M15 gene, which encodes an extended spectrum beta-lactamase conferring resistance to ceftriaxone, and it carries a transfer locus that allows self-transmission of this plasmid to other Salmonella typhi and Paratyphi lineages. Now, if this wasn't bad enough that it had all these resistances, this happens to be the strain that has the ability to displace endemic strains in your community. So if I get on a plane and I'm Typhoid Mike and I travel to your city and I make you the episode shouldn't be called Typhoid Mike. And I
was thinking about it. It's a good one.
I know. and so if I travel to your city and I'm making food for friends at a house and I'm actually shedding it, it can actually spread very readily in the community. And what this research group learned is from the Public Health Service of the United Kingdom, one of these isolates from Pakistan has already made it to Britain.
Not surprising. There's a lot of traffic back and forth between those countries, right?
No. And so you just get the sense, just looking at the data, this is not good. And in fact, the collective results and the term they use is panoply of chromosomal and plasmid antimicrobial resistance genes. It is that this outbreak in Pakistan with this organism encodes resistance to all main antimicrobials that have been considered first-line drugs to treat typhoid fever during the past 70 years. And if, when we look at this, you can now see, and this is where I'm going to bring in Typhoid Mary, because Typhoid Mary lived between 1869 and 1938. And this is the success story of shoe leather epidemiology. She was the first person identified as an asymptomatic carrier of this pathogen. She's thought to have infected 51 people who she was cooking for, 3 of whom died over the course of her career. And it was, shoe leather epidemiology promulgated by George Soper, who effectively figured it out. And he ended up incarcerating or quarantining Mary between 1907 and 1910. Now, this woman had committed no crime other than cook, and she transferred this organism to these people. And like some politicians in North Carolina that we talked about once, she didn't understand hand hygiene, nor would she consent to having her gallbladder removed. And then under New York City Code, she was quarantined. And that's New York City Code 1169 and 1170. And she was literally in isolation on the North Brother Island, which is it's in the East River. Vincent probably can see it from his office. And she was there for 3 years. Eventually, Eugene Porter, who was the New York Public Health Commissioner for the state, let her out. She swore blind she would never cook. She took a job as a laundress, but she lied, changed her name, and she returned to cooking. And so then they began the hunt to quarantine her for the second time.
Wow.
And, Mary is literally 100 years ago. And Clem and colleagues are saying, okay, folks, we may not have someone as famous as Typhoid Mary, but Clem and colleagues employed many of the same techniques in the last century during this outbreak, albeit with very sophisticated molecular tools. And you get the sense of outbreak tracing when you look at their remarkable first figure, and they show you the XDR, the extremely drug-resistant samples, are separated by 6 single nucleotide polymorphisms that rest on this blue branch. So it's, literally tracking where these microbes came from. And it's just absolutely frightening. And they conclude their call to action by matter-of-factly stating the emergence of spread of XDR typhi in Pakistan and the startling demonstration of how ubiquitous antibiotic resistance plasmids can be acquired by the MDR Salmonella typhi, rendering it extensively drug-resistant, narrowing our treatment options, really is a call to action that we really need to think about investing in sanitation, namely sewage treatment plants, as well as water distribution and again, reinforcing the importance of hand hygiene in the food preparation areas. Now, I know there are some individuals out there arguing about small government, and we don't need to tell people they need to wash their hands, but we really do. Now, I hate to end on that bad note, but fortunately, there are vaccines available. There are two. There's a heat-killed, and there's a live attenuated vaccine. And in the show notes, I'll put the CDC's recommendations on the vaccine and dosings. Unfortunately, the vaccines don't seem to be very effective for very long, but they will protect you if you're traveling to areas endemic. So I think in addition to the shot heard around the world, it is really dropping the gauntlet to really improve the efficacy of the two licensed vaccines that are out there. And I have the greatest hope for the oral typhoid 21A vaccine because I think things are getting much better. So the bottom line is wash your hands, drink clean water, and hope if you eat out, the cooks and food servers are not carriers, or even if they are, they wash their hands so as not to contaminate your food. Even if less government types don't think we should have to legislate hand washing. So that's the snippet.
Boy, that's pretty grim. Now, the total drug-resistant TB, has that spread globally?
No, and I think it's because it grows so damn slow.
How about this DR? Does this grow slowly? Apparently not, right? It can
displace. No, this is a God-fearing enteric. It has generation times measured in minutes.
God-fearing enteric. That's great.
Yeah, it's out there. And the thing I think that's really remarkable is that for many years, Salmonella had this incompatibility group plasmid that was keeping the resistance plasmids out of it. But then as we transition towards the beginning of this century, the R plasmids, the resistance plasmids that have these transposons, figured out how to stay inside of Salmonella. They could always get in there, but they would be quickly kicked out. But now they figured out how to stay stably maintained. And, the only thing left that we have is now we go to the things like the carbapenemases or some of the more expensive drugs. And those things aren't administered orally. They're administered IV. So even for us folks in the developed world, it's not going to be a cheap disease to treat because you're going to need to be hospitalized or go to an infusion center in order to get the drugs.
I think part of the problem is that antimicrobials were just too easy and a lot easier than cleaning up, right? And not getting fecal contaminated water. But that's really the key. You need to clean up and not dump feces into drinking water. And it's really hard.
That's what worries me. The Clean Water Act was end of the 60s, beginning of the 70s. And, that's been 50 years ago that we had a substantial influx of capital to improve our sewage treatment plants in the country, move off of septic, and to make certain that we had good potable water being delivered to every household. So I think our politicians need to think about infrastructure and, simple things like water and sewage treatment are big ones that I think we need to worry about again.
You better tell the EPA because they don't seem to care.
They will if they get Salmonella typhi because it's not a pleasant disease to have.
Have you had it?
No, but I've had diarrheal diseases where I, was bent over with gastric pain. I
have too. I went to France once and I had something which was horrid.
Yeah.
I don't know what it was, but it lasted about a week. Foodborne. I think I got it on the plane.
That was bucket number 2. So those are the ones where it's a self-limiting infection.
Yeah, it doesn't go systemic, right?
Yeah.
I learned a lot, Michael. Thank you.
Well, you're welcome.
I love the God-fearing enterics. That's great.
Well, Typhoid Michael is.
Now we won't use that, but it's a nice phrase. However, you haven't had typhoid, so you're not.
No.
Elio, did you ever have typhoid?
No, I haven't. Although I lived in a country that had plenty of it.
Yeah. All right. Thank you, Michael.
Thank you.
I want to tell you about the upcoming ASM microbe. the annual meeting of the American Society for Microbiology. Anyway, this year it's in Atlanta, Georgia, and ASM has a special opportunity for our podcast listeners. Get $50 off registration for Microbe 2018, which is June 7th through 11th in Atlanta, using the promo code ASMPOD. ASM Microbe 2018 connects scientists with their science and showcases the best microbial scientists in the world. Delve into your scientific niche in 8 different tracks. Don't miss this opportunity. Visit asm.org/microbe. That's asm.org/microbe and use the promo code ASMPOD. It's all one word for $50 off registration. When you go to register, you will see, do you have a promo code? You type in ASMPOD. Now, for something completely different, we are going to talk about amphibians and fungi. And this fungi are one of Elio's favorite things. And everyone likes a nice frog, right?
They're cute.
Cute. And this is a paper published in Science in March of 2018. It's called Shifts in Disease Dynamics in a Tropical Amphibian Assemblage Are Not Due to Pathogen Attenuation. And the first author is Jamie Voiles, and the last author is Corinne Richards-Zawacki. They're from University of Nevada, University of Massachusetts in Boston, the Smithsonian Tropical Research Institute in Panama, University of Pittsburgh, UC Berkeley, New Mexico Institute of Mining and Technology, Arizona Game and Fish Department, Vanderbilt University, and a few other places in Panama.
A couple of field stations in Panama.
Yeah, a few field stations in Panama, exactly.
In the canal, probably.
No, this map here, I'm not sure where the canal is, but there are 3 sites in Panama. And this is all about the fungus that is wiping out amphibians globally. It is the chytrid fungi, chytridiosis. And we've talked about this recently on TWiM, Batrachotrichium, right? Batrachochytrium salamandrivorans. That's the one causing a collapse of the populations of salamanders in Europe. And the other one is Batrachochytrium dendrobatitis. This is the one causing the lethal skin disease of frogs. These are fungi. It's a chytrid fungus. It's the phylum Chytridomycota. There are about 1,000 different chytrid species. They live in water or moist environments, among the oldest kinds of fungi. And as I said, they're causing global declines of amphibian species. They're spread by the amphibian trade. You know, you move frogs around the world because you want one and it's cute, and you introduce the fungus into a population that's never seen it before, and the population collapses. And this is a skin disease, as I said. And in this paper, they're going to use this, in particular, an outbreak in Panama, to understand how do infectious disease outbreaks end. So typically you have an outbreak, there's an epidemic or epizootic phase, and then you transition to a phase where the hosts and the pathogens coexist. This is the endemic or the enzootic phase. They want to know what are the mechanisms that underlie this transition. And, you know, typically this involves not just the pathogen, but the host and the environment. So it's a little tricky. But this system, which they use to study this question, is interesting because you have the frogs before and after the epidemic, and you have the fungus before and after the epidemic. So you can do all the infections. Of course, you can't do that with people. You know, when people outbreaks end, but you can't get people and reinfect them and see what's going on. So we have to have animal models like this. So that's the cool part of this study, is that they are using this, you know, decline of this amphibian population to address this question. So the story is that over 10 years ago, there was a series of amphibian declines at 3 sites in Panama, and these have been well studied. They are El Cope, El Valle, and Campana. And during these outbreaks, they saw increases. What's that?
El Valle.
El Valle. Thank you. During this outbreak, they saw increases. I'm going to abbreviate the chytrid fungus as BD.
Called BD. Thank you.
Okay. Becton Dickinson.
Yeah,
BD.
No, you're going to get letters for that.
BD. So the BD levels went up. The amphibian densities went down. And these 3 sites have been studied for a long time by field surveys. So they have a lot of historical data. But between 5 and 13 years after the original outbreak, some of the frogs began to recover, but BD was still around. So the idea was it was a epidemic, and then there was a shift to an enzootic phase where the frogs and the fungi are coexisting. So they want to know what's going on here. And they say, well, here are the possibilities. The pathogen lost virulence, maybe the host resistance increased, or maybe both. So they do a bunch of studies in this paper to try and get at this. And this is a quaint little paper. They call their work garden variety experiments.
I love that.
Actually, they say garden variety and genome sequencing. So the genome sequencing is not garden variety, but everything else is. And, a lot of the paper is written, if you read the methods, it's all we did this, we did that. It's very different. So they have 12 species that live near the riverbanks, right? And they were nearly wiped out by BD. And they have a graph showing the pre-outbreak numbers of these frogs. They have not 12 different species. You can see they went way down during the outbreak, and then some of them are coming back again. And what are these? These are these interesting frogs like the harlequin frog, the rocket frog, these very interesting, colorful frogs. And now the composition of this frog community is sort of what it was, or similar to what it was before disease broke out. So, that's a good thing, because we're afraid that these fungal outbreaks are going to wipe out amphibians. I think over 100 species have already been extincted by this. So understanding what's going on is important. Okay, next. So that was one part of the paper. They did a survey of what frogs are still there. The next thing they did is they sampled the frogs. They take skin swabs from these animals, because that's where the fungus is, on the skin. And then they look for it by PCR. So they collected 2,035 samples, and they found that the prevalence of BD has gone down because they have some samples from pre-epidemic phase and during the epidemic, which they can compare from the literature. And they see that the BD has gone down. Just the number of frogs that are infected is much less. Okay, so that's part one. Next, how about the pathogenicity of BD? Has it changed over the years? So they have some isolates from 2004 during the epidemic. They call those historic isolates. And then they have isolates of the fungus from 2012 to 2013. This is after the population has recovered. They call this contemporary isolates. They have 3 of each. They measure the growth of the fungus, the size of the spore case, which is the zoosporangium, and the numbers of infectious zoospores, which are the spores. These are motile asexual spores that have flagella. No differences between historic and contemporary samples. Okay. Next, a big part of the growth of the fungus is the ability to evade host defense mechanisms. We know that the skin of these frogs have antimicrobial substances that will kill the fungus. So they ask, well, is there any difference in the ability of the fungus to grow in the presence of this? So they get skin secretions, actually get either skin secretions or the supernatants of 18 different skin bacteria. These are bacteria from the frog skin that are known to inhibit BD. And they add it to BD samples and assay growth, and there's no difference in the inhibition comparing the old ones to the new ones. So the fungi have not changed in their ability to grow in the presence of these skin antimicrobials. The fungi also make factors that kill B and T cells. And that's part of the reason why they're so pathogenic. So what they do is the They take the supernatants of these fungal strains, they add it to T and B cells, and they measure proliferation. No difference. So the old strains of fungus, and the new ones, they both kill B and T cells equally well. So there's been no change there either. Are we okay so far?
Yeah, and it's reminiscent of how it's not the same, but it's reminiscent of HIV in humans. You know, HIV kills our immune cell, and it could potentially be species ending as well if it gets out of control. And so this paper is interesting because it helps you think about what's going to long term going to happen with HIV and the human dynamic. And if we were lucky, we'd be through of HIV, but we're not as lucky as the frogs.
Well, here we can do experiments that we can't do in people. That's the beauty of this, right? So you can then you can try and learn something that you can apply. I was thinking,
is it possible to think back historically about diseases which have disappeared? Because if this, if you carry this to a logical conclusion, resistance by the host may render the pathogen ineffective or get rid of it. So I'm wondering, I don't think I know of any cases like that where the disappearance of the disease wasn't due to immunization or antimicrobials. I wonder if you can you think of something?
Yeah, I thought the same. That's a good question. I thought the same thing. I can't think of any viruses that have gone away. What about bacteria?
But, if you were a clinical microbiologist in the year 1000, you would probably be isolating bugs which you never heard of today.
Yes, of course. We don't have that historical data, right? And
I guess the only thing that would immediately come to my mind is the incidences of scarlet fever, because that has waned. But that comes on a lysogenic phage, and that's just phage dynamics of being excluded from the population. And antimicrobials have, of course, gotten rid of those organisms or quickly depleted them before they could spread.
But the point is that biology being what it is and evolution being what it is, some diseases will disappear because the host will adapt to them, just like the frogs did.
Yeah,
so the time frame is enormous, and in humans it's very, very long. But it will happen.
Yeah, no, I agree. But now we have in humans anyway, we have the complication that we intervene, right? So
we tinker.
We tinker, so it's hard to know what would happen otherwise. But here we have a population where we haven't tinkered yet, and we can look, and they do rebound in some cases, and so that's interesting to look at. But really, in the end, as you'll see, it's all about immunity. All right, the next experiment, they said, has the pathogenicity of BD changed? And they get 2 different frog species. Now, this is where I worry a little bit about the results. They get 2 different frogs that they can buy, okay? One of them is Atelopus varius, which is descended from frogs that are near El Cope, which is one of these sites, in 2002, before the emergence of BD, right? So this is supposed to represent what's there before the outbreak, and I guess that's okay. It's the best you can do. And they infect them, and they can put them in baths with spores in them and get them infected. And then they compare the old and the new BD isolates, and there's really no difference in their ability to infect this frog. The other frog they use is Litoria Cereulia, and this is a common frog. I'm not sure it's a common green tree frog. I don't know if this frog is present in the area, but this is purchased also, so I don't know if that's relevant. Again, there's no difference in the 2 isolates, but I just think it would be better to have frogs completely captured from the site, but I suppose that would be hard because they would be BD positive and so forth. And they also did transmission experiments. They would infect the frog, put it in a bath with another frog, and see transmission. There's no difference between the historic and the contemporary BD isolates. They sequenced the genomes of these fungi, the old and the new fungi, whole genome sequencing. They looked at 25,000 single nucleotide polymorphisms. They say nothing stood out as being really different when you compare the old ones and the new ones. And that's not garden variety, by the way. That's deep sequencing.
Mm-hmm.
And the last experiment,
It was deep.
in the last experiment, they looked to see if there's any differences in the frog's ability to defend themselves against infection. So they take skin secretions and they have hosts pre and post epidemic, and they incubate it with the fungus, and they see differences. This is the first time they see a difference in this paper. There's more, and they have a lovely graph where they, unfortunately, you won't be able to see it, but I'll tell you what it says. If you compare pre and post disease frogs, you see that the frogs they differ in their ability to inhibit the fungus. Okay, so they have this graph showing pre-disease and post-disease inhibitory effectiveness. So frogs pre-disease don't have a lot of ability to, their skin secretions don't have a good ability to inhibit the fungus, but frogs post-epidemic have a pretty good ability to inhibit the fungal growth. And in fact, they did a cool experiment where they compare the skin secretions of captive frogs with that of wild frogs, and the captive frog skin secretions don't really inhibit the fungus at all, whereas the wild frog skin secretions do. So they think that there has been some shift in host resistance of the frogs, and that's why they're recovering, right? Because the fungus hasn't apparently changed, but the frogs are recovering, and they think it's because of resistance, maybe in part from the skin secretions. And, you know, the skin has antimicrobial peptides that are produced by the microbiome, right? So it would be interesting to look at that and see what's going on. I suspect there's also something inside the frog that's happening as well, like adaptive immunity that has been slightly different. So, and then the final question is...
Did I hear you right? You said that the antimicrobial peptides are all made by the microbiome?
No.
I wasn't aware of that. Is that the case?
Yeah, most of them
made our own.
No, most of them are made by bacteria that are on your skin.
Stuck to the skin.
Yeah.
Defensins and cathelicidins are made by us, not by bugs.
Yeah, some are made by us, but some are made by bugs. No,
right.
The magnanins.
The first one known by Dubose, gramicidin.
Antimicrobial peptides are part of the innate immune response. Yes, okay. Bacteria. Let's see. Some are made by bacteria. I'm pretty sure. Maybe not all of them.
They are, but I think the most important ones to our body are made
by us.
You know, the granules of granulocytes, full of it.
Yeah. So
anyhow,
so they were originally called bacteria. They were originally called bacteriocins, right?
Yes,
because they stopped. Right. Yeah. Okay. Yeah.
Okay.
So it would be interesting to sort that out. Now here's a good point. Oh, remember the frogs were wiped out by the fungus, and then they returned. Where did those frogs come from? They didn't just spontaneously generate, right?
No.
So they think they came from somewhere else, from lowland areas, right? But the thing is, they've looked at those frogs, and they don't have skin secretions that are resistant to the fungus. So that doesn't quite make sense. I think, and they suggest this also, that there were a few frogs left. They didn't catch them all. They missed a few, you know.
Or the fungus missed a few.
Yeah, exactly.
Because it didn't crash the entire population.
Or maybe there were a few resistant frogs that were infected, and they left. They were left over, and then they reproduced, and that's what we're seeing nowadays. Because I'm not sure they could get every last frog right during the epidemic. Who knows? Either way, there's something to do with immunity, which is good because it tells us if we can figure out what it is, maybe we can help some of the populations that are crashing now. And I was looking around the literature, and other people have been studying, you know, the kinds of immune responses to this fungus. You know, it's more than just a skin secretion. So I think it's more complicated than they make it. So the bottom line is, it looks like the frogs have become somewhat resistant to the fungus. The fungus hasn't changed. And in this particular sites, the frogs are coming back, which is good. And so maybe we can learn something and help frogs elsewhere.
Well, I think it's a great food for thought, and one has to think about the evolution of infectious diseases in all systems, including ourselves. Obviously, having an animal with a very short lifespan and very short life cycle is easier to handle than humans, where the time scale is so much longer.
Yeah, so this is a 10 years, evolution, and that would probably be much longer in humans, right? It would be hundreds of years to see a disease disappearing. So, yeah, that's a good point that you can have a manageable study here. And I should note, funding for this study was provided by several sources, including the Disney Worldwide Conservation Fund. Disney loves cute animals, and they're supporting research on them. Anyway, there you go, frogs. And so that's, we've done a couple of fungus papers lately, so it's pretty
nice.
Good. It's pretty good.
Good.
All right, now I want to end up with giving away a book. We're giving away Management of Clinical Microbiology Lab from ASM Press. And I told people to send an email with micromanagement in the title. And how many people did we get here? We got 13 emails. We got one from Rachel, from Anthony, from Katie. Now, Katie says, I love the podcast, but as a microbiologist, I feel that the fungi are left out. But here today, you had a fungus. There you go, Katie. Joseph is a lab manager at Princeton University, where he makes plates for the lab. Loves to listen. Sabrina is a microbiologist in a clinical lab in a small community hospital. Stephanie has worked in a clinical lab. She loves all the TWiX podcasts. John is a devoted listener, a third-year microbiology student who is currently procrastinating by listening to your podcasts. He worked in a summer clinical microbiology lab at Linköping, at a university hospital in Linköping, Sweden. And he says, while I'm writing, I might as well mention my
Linköping.
Linköping. Linköping. It's not Linköping, huh? Okay. While I'm writing.
It's that over the O.
Okay, thank you. I want to mention my fascination for the symbiotic relationship between the stingless bee and the fungi you covered in 171. And there's another fungus episode. My grandfather is a beekeeper and has over the years raised concern about beehives being lost without explanation. It would therefore be very interesting to investigate first if there is a similar relationship in honeybees. If that's the case, one could follow up with the effect of various chemicals used in farming on the growth of fungi and consequently the bees. Absolutely. We got an email from Megan, who works in a microbiology diagnostics lab. One from Nadia, who is her future, she said, is clinical laboratory management. That's her chosen career path. She also writes, Michelle Swanson and I were at the National Postdoc Conference this weekend in Cleveland. I recognized her voice. I wanted to say hi. Met her in Ann Arbor, but didn't meet her. And it was great that she came to this postdoc conference. Us postdocs need faculty advocates, and I very much appreciate any mentor that makes efforts to improve postdoc training. Oifi writes, hey, Elio, how would you pronounce the word or the name AOIFE?
AOIFE. I don't know.
It's Irish. Oif, Eve.
Maybe Eve.
Eve. Anyway, he or she is in second year of college studying science in Ireland. And Larry wrote, Amanda is a TWiV listener. She's finishing up a clinical microbiology fellowship in Halifax, Nova Scotia, and then will become the Saskatchewan Provincial Lab Director of Virology and Immunoserology. The book would be a welcome addition. And finally, we have Adam is from San Francisco. He's a clinical lab scientist doing microbiome work. And Philip is he got his BS in microbiology from the University of Arizona many years ago and has since moved into clinical lab science. You all do such a great job explaining the different papers, from the design of the study to the quality of the data, and present complex techniques in such a way that even I understand them. I love that you always include potential applications of all the papers, many of which apply to my field of medical microbiology. We are always looking for ways to identify organisms and antimicrobial resistance faster, so I am always curious what techniques will jump out of research labs and into hospitals. For example, after agar plates dominated clinical lab identification for decades, MALDI-TOF has completely changed hospital lab workflows, and I expect further improvements to come at a rapid pace. Thank you all for your time and effort. I haven't been this excited about reading research papers in a long time, maybe ever. So this means your show is amazing. Thank you, Phil.
And I've figured out how to pronounce IFA. That is how you pronounce it. And
IFA?
IFA. And it means beautiful, radiant, and joyful. Thanks to our good friends at Google. And it's an
Thank you, Michael.
Irish name.
That's very nice,
It's an
Michael.
Irish girl's name.
It's an Irish girl's name. All right, so I just picked a winner here. The random number is number 8. That's Nadia.
That's Nadia.
So she is the winner. Nadia, send me your address to TWiM@microbe.tv, and we'll ship out this book to you. And congratulations. And for those of you who did not win, there will be other opportunities to have books of different sorts. And that'll do it for TWiM 174. You can find it at asm.org/TWiM. You can find it on your favorite podcast player. Please subscribe. And if you like what we do, you can become a patron and donate. Go to microbe.tv/contribute for ways that you can do that. And of course, we love getting your questions and comments. Send them to twim@microbe.tv. Elio Schaechter is at the blog Small Things Considered. Thanks, Elio.
My pleasure. Thank you.
Michael Schmidt is at the Medical University of South Carolina, the harbinger of bad news this week, Michael.
I'm the harbinger of bad news.
Thank you, Michael.
Yes. Thank you, Vincent. Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, Ray Ortega for technical help, and Ronald Jenkins for his music. RonaldJenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.